Chapter 12, Biotechnology and its Applications. In that, 12.2, Biotechnological Applications in Medicine. There are a lot of scope of biotechnological applications that present in the medicine that has taken into very new heights in the field of medicine. The recombinant DNA technological process have made immense impact in the area of healthcare by enabling mass production of safe and more effective therapeutic drugs. The recombinant therapeutics do not induce unwanted immunological responses, as in common in case of similar products isolated from non-human sources. In 12.2.1, genetically engineered insulin. So insulin, we all know that it's an important regulatory substances that will control the blood sugar level. Nowadays, in most of the people, diabetes is the physiological failure of one's body that the person cannot maintain the blood sugar level because of many factors like hereditary factor, lifestyles, and less work, more carbohydrate diet. There are many causes of the onset of diabetes. Early age people, say from 30 years, starting from 30 years, the people are more susceptible to the diabetic diseases. So they cannot control the blood sugar level inside their body. So in a very previous days, the insulin that will regulate the level of blood sugar that was extracted from the pig or from the pancreas of the calf. So even though they are more similar, but they cause some immunological problems. So they cause some allergy. So to overcome most of the problems, we have genetically engineered the insulin production protocol so that mass production can be done. Say here in textbook it says insulin used for diabetics were earlier extracted from pancreas of slaughtered pigs and cattle. Insulin from an animal source though cause some patient to develop allergy or other types of reactions to the foreign protein, because it is a general thing that the foreign protein that inside the blood will trigger the immune reactions that results in complications. Insulin consists of two short polypeptide chains. Say this is two short chains, A peptide and B peptide. They are linked together by the disulfide bridges. In mammals, as a human, insulin is synthesized as a prohormone. So not in active form, it was synthesized as a prohormone, like proenzyme. The prohormone also needs to be processed before it becomes a fully mature and functional hormone. This is a proinsulin, this is a prohormone. It has to be processed before it should get activated. Otherwise, it won't activate it. So why this is? Because it has another chain that is known as peptide, another chain, the C chain, C peptide chain that is attached to the A chain and also this B chain. This has to be processed. The C chain has to be removed to make the insulin an active hormone, which causes an extra stage called C peptide. The C peptide is not present in the mature insulin. So this we are more concentrated on the mature insulin where this is the functional. The main challenges for production of insulin using recombinant DNA technology was getting insulin assembled into a mature form. It's very difficult. If we clone the human gene and we process, then we get the proinsulin. but it is very difficult to get the mature insulin. So, in 1983, an American company, Eli Lilly, prepared two DNA sequences corresponding to A and B chain. What they have done, they have separately prepared these A chain and B chain separately in two separate genetically engineered plasmids, and they extracted and combined by creating disulfide bonds to produce human insulin or humulin. So what they have done, what this company have done, very beautiful mechanism. They have taken the bacteria, E. coli bacteria, with this plasmid containing the gene that produces only the A polypeptide, human gene that produces only the A polypeptide chain, A chain of the insulin, and they have took another set like a B chain. They separated, they extracted from the downstreaming process, and these two, the bioproducts like A chain and B chain, they mixed, and the formation of disulfide bridges between these two A and B chain that resulted in the functional insulin. So this is a very beautiful technique of making the mass production of the purified form of the insulin, that is saving many of the people who are suffering from the disease diabetes. So it is a very beautiful gift by the biotechnological process. Next, another is gene therapy. If a person is born with a hereditary disease, can a corrective therapy be taken? So, gene therapy is a collection of methods that allows correction of gene defect that has been diagnosed in child or embryo. So here, genes are inserted into a person's cells and tissues to treat a disease. Correction of a genetic defect involves delivery of a normal gene into the individual or embryo to take over. So the correct gene has to be introduced to the embryo or to the any of these stem cells like bone marrow to take over the function and compensate for the non-functional gene. If the patient doesn't have any functional gene, the genetically engineered tissue that containing the functional gene can be reintroduced so that the newly introduced gene that will take the function of the non-functional gene, so that the patient can survive. The first clinical gene therapy was given in 1990 to a 4-year-old girl with ADA deficiency, adenosine deaminase deficiency. This enzyme is crucial for the immune system to function. So this ADA is necessary for the lymphocytes to get mature, so that they can active against any other antigens. So the patient can fight against, the person can fight against the disease. In the absence of this ADA, so the immune system doesn't function properly. This order is caused due to the deletion of a gene for adenosine deaminase. Say the chromosome 20 is involved in this. In some children, ADA deficiency can be cured by bone marrow transplantation. So the bone marrow contains the hematopoietic stem cells. So they are directly derived from the embryonic stages. So where the complete replacement of this bone marrow tissue, they may be helpful in the correction of this particular problem. In others, it can be treated by enzyme replacement therapy. In some, enzyme replacement therapy can also be functional, in which functional ADA is given to the patient by injection. A functional ADA enzyme can be injected in the patient. But the problem with both of these approaches that they are not completely curative. They are just temporary. They are not completely curative. As a first step towards gene therapy, lymphocytes from the blood of the patient are grown in culture outside the body. So the lymphocytes of the patient, they are taken out in vitro, they are grown on the special medium, and it is infected with a retroviral vector containing the functional ADA copy DNA. So the retroviral vector containing the functional ADA gene into the isolated lymphocyte cells of the patient. So we are loading with a functional gene is then introduced to these lymphocytes, which are subsequently returned to the patient. Then those cells, they are replaced to the patient's bone marrow cells. However, as these cells are non-immortal, so they cannot live for very longer because they are not actually stem cells. The patient requires periodic infusion of such genetically engineered lymphocytes. So for that, the patients has to get again and again these lymphocytes at regular intervals for the proper functioning of this gene, ADA gene. He has to take the functional ADA gene for the proper functioning of his immune system. So, however, if the gene isolated from bone marrow cells producing ADA is introduced into the cells at early embryonic stages, it could be a permanent cure. So, if we detect the problem, this is genetic disease, if we detect the problem at the embryonic stage itself with many other techniques, so that we can include these genes into the embryonic cells, so that it will be promising for the permanent cure.